discuss a little bit of my research regarding vitiligo, which is an autoimmune disorder. So vitiligo happens due to the loss of melanocytes from the skin. Melanocytes, these are the specific cells which synthesize a pigment known as melanin. And you see that wherever your melanocytes are lost, you will see no melanin is there, and the patches, white patches, you can observe you. Okay, so the disease is you can say very much crucial for the people who are having it, because of these people may have certain matrimonial problems, they may have self-less esteem, and social stigma as well. So these are the melanocytes which are residing in the basal layer of the epidermis, and these are also dendritic cells, and melanin is synthesized in the specialized compartment known as melanosomes. These melanosomes will then take the melanin up into the upper surface of the skin. So somehow these melanocytes are getting died. Okay. So what are those processes? What are those immune alterations? Whether this melanocytes they are able to present MHC molecules or not properly, that we are going to see. So why there is a death of melanocytes? Because somehow these melanocytes they are releasing their antigens, and these antigens are recognized as autoantigens by our own immune cells. Okay, so this is the humoral immunity which has been imparted by your B cells. We all know that B cells produces antibodies. So these B cells, you can see that they are recognizing these melanocyte antigens, and it gets activated and produce lots of anti-melanocyte antibodies. Okay, so anti-melanocyte antibodies can initiate a pathway known as antibody-dependent cell-mediated cytotoxicity. Okay, they can also activate complement fixation of complement-mediated pathways, which can cleave this particular melanocyte. The melanocyte antigens can also be taken up by the antigen-presenting cell. As I just told you that antigen-presenting cell, they have MHC class 2 molecule as well as MHC class 1 molecule. So when these antigens are taken up by MHC class 2 molecules, they present the antigens to the T helper cell, and T helper cell will then get activate these cytotoxic T cells and B cells as well. And this happens through several cytokines. Okay. Cytotoxic T cells alone can also recognize the melanocyte antigens presented by MHC class molecules by these APCs. Okay, so cytotoxic T cells will then again activate and try to release certain cytotoxic compounds which will cleave these melanocytes. So one thing is clear that yes, B cells are involved in clearing out the melanocytes, T cells are involved, as well as antigen presenting cells are involved. Right? But the question lies is here is that what triggers the melanocytes to die or to release their own antigens? So let me tell you that the triggering factors or triggering agents are not yet classified, but the role of physical and chemical stresses are emphasized. Defective apoptosis, that is a programmed cell death, when it gets early, or any defects in the apoptosis can also lead to the melanocyte antigens to appear in the blood. Altered self-antigens. Sometimes our own body cells will try to exhibit our antigens, self-antigens differently. Okay, so then these are recognized as non-self. Okay, by our immune system. Sometimes neoantigens will be formed during the post-translational modifications of proteins. So that may also trigger this type of inappropriate immune response. And obviously heat shock proteins like HSP70 and all, they are very important to present the antigen by antigen presenting cells. So here in this figure I am trying to show you that how melanocytes are releasing its antigens and what are these antigens. Okay, so you see that melanocytes, they interact with the Langerhans cells. So these Langerhans cells, they are present in the skin, right? So this communication will lead to the apoptosis of the melanocyte. So melanocyte apoptosis will release the melanocyte antigens like PMEL17, MART, tyrosine related protein 2, protein 1, and tyrosinase enzyme. These proteins are essentially involved in the melanogenesis process. Okay, but in research we have found out that they act as potential autoantigens, right? So these antigens will be taken up by dendritic cells. They present these antigens to the T helper cell. T helper cell can activate, you can say, B cells, and the B cells will release anti-melanocyte antibodies that will kill the melanocyte. T cells can also activate cytotoxic T cells with the help of interleukin 2 cytokines. These cytotoxic T cells can again directly kill the melanocytes. So this is the pathway at how autoantigens or the cells of melanocyte antigens, they can be reacted and destroyed by the immune cells. So these are some of the data of my lab that we have shown that anti-melanocyte antibody levels are quite high in vitiligo patient as compared to control. Okay, and there are two types of vitiligo known as generalized vitiligo and localized vitiligo. So we have seen that generalized vitiligo is more associated with high levels of anti-melanocyte antibodies, and it has been seen that in generalized vitiligo you will see more of autoimmunity. We have also checked the anti-melanocyte antibody depending upon the progression of the disease. So active vitiligo patients, those who have recent vitiligo, we found that anti-melanocyte antibodies are quite high. In the female patient, we have seen that these anti-melanocyte antibodies are quite high, and as well as in the early age of onset of the disease. So you see that the age of onset 1 to 20 years, you will find more of anti-melanocyte antibodies as compared to the late age of onset. Now to determine whether this anti-melanocyte antibodies comes first or the other triggering agents they are coming first. So we did this experiment to show that there is an oxidative stress which is been you can say I think someone mic is on. Hello, someone mic is on. Yeah. So can you switch off? Yeah. Okay. So lipid peroxidation is a marker of oxidative stress. We have found that the patient who have recently developed vitiligo in the past 3 months, they exhibit lots of lipid peroxidation levels as compared to the late age of onset of patient. But the anti-melanocyte antibodies was found in late age of onset patient, but they are not found in much larger quantities in the less than 3 months. So this suggests that your oxidative stress comes first as a triggering agent, right? So that the melanocyte antigens are released and then anti-melanocyte antibodies are synthesized and their levels are rising. This table provides a list of certain melanocyte autoantigens against which antibodies and T cell receptors are reacting. So it includes tyrosinase related protein, female 17, it is also known as glycoprotein 100. You have MART1, melanocortin hormone receptors, SOX9 and SOX10. These are the proteins which are involved in the embryonic development. So all these have been suggested to be a potential autoantigen against which the autoimmunity develops. So almost 90% of the patient you will see that anti-melanocyte antibodies are there, and these antibodies are related to the IgG1 subclass 1, 2 and 3. The another important role of IgG antibody in addition to the ADCC is that IgG antibody can allow the increased expression of ICAM1 molecule. Now this is known as intercellular addition molecule 1. So it makes close proximity to the T cell. So whenever this ICAM1 is expressed in high amount on the melanocytes can get readily attached to the cell for the destruction. Also, IgG can lead to the increased production of interleukin. Reported that interferon gamma can actually induce ICAM1 expression. Okay, so we found that interferon gamma levels in vitiligo patients were high, so that may lead to the increased ICAM1 expression, and that's how we can suggest that it may be associated with the melanocyte destruction. Later, a few years back, we have published one genetic association study related to the LMP7. LMP7 is also called as proteasome subunit beta 8. So if you remember that in the cytosolic pathway, I told you about LMP2 and LMP7, which are involved in the processing of your endogenous antigens. So what we have observed in the vitiligo patient is that this PSMB8 polymorphism is associated with the vitiligo patient, and that polymorphism is also associated with the less expression of LMP7. Okay, so we have found that specific CC genotype leads to a decrease transcript level of PSMB8. Okay, so we have monitored both transcription level as well as protein levels. So you see that here in the patient, the PSMB8 mRNA levels are quite low as compared to controls. And if you go along with the genotype, the patients which are harboring PC, they are having very less expression of PSMB8. Okay, so if PSMB8 expression or LMP7 expression is less, then it can lead to suppression of MSC class 1 molecules. Okay, so melanocytes will not have sufficient MSC class 1 molecule on their cell surface. So this will be just or checked by your cytotoxic T cells. It will be judged by your nature killer cells. Okay, so they will find it defective and then they will try to kill the melanocytes. So as I just told you that protein expression is also less. You can see the in control, this is the Western blot analysis. You can see that the PSMB8, which is a 21 kilodalton protein, in controls it is quite high, but in patient it is quite low. Now one thing is clear that yes, the melanocytes are getting destroyed by cytotoxic T cells, right? What are the natural mechanisms which are present in our body that will control this? Okay, because nature has provided us that whenever our immune system got hyperactivated, it must be suppressed to a normal level. So that suppression will be done by regulatory T cells. Okay, so in vitiligo patients, we have found that the regulatory T cells function and number is also decreased. So that's how you have unchecked cytotoxic T cells, which are continuously killing or destroying these melanocytes. So these melanocytes in cell culture will look like this long dendrites containing cell. So let's understand that how cytotoxic T cells, by which mechanism it will kill the melanocyte. So you see that cytotoxic T cells is recognizing the anti-melanocyte antigens, right, autoantigens. So cytotoxic T cells will now release granzymes and perforins. So perforins are actually the pore forming proteins. You see that these blue color structures, they are forming a channel of pores and through which the granzymes can be entered. Okay, so perforins will create pores and granzymes will follow it. Okay, once granzymes reaches into the cytoplasm, these granzymes will act as esterase enzymes. Okay, so granzymes will then activate the caspase pathway. So this is a program cell death pathway in which caspase enzyme plays a very important role. And those melanocytes which has caspases activated, they will undergo cell death. So this is how cytotoxic T cells will lead to the melanocyte cell death. NK cells also do surveillance of our each and every cell of our body. Okay, MSC molecules are not just important for humoral and cellular immunity or to present the antigen, but it also do self versus non-self recognition. Okay, MSC molecules are identity card of our cells. So whenever insufficient MSC expression is there or any structural defect is there in the MSC molecule, NK cells with receptor known as KIR, that is killer cell immunoglobulin-like receptor, it will recognize those defects and it will kill those abnormal cells. What you see here in this picture is that this NK cell is recognizing these MSC molecules correctly, so that this is giving a negative signal inside that do not kill this normal cell. But when a cell is expressing very less MSC molecules, or let's say a defective MSC molecules, this KIR receptor cannot bind to the NK cell MSC, means it is not recognizing. So this will give a positive signal inside the NK cells, and NK cells will release cytotoxic compounds so that this type of abnormal cells will be killed. So as I told you that cytotoxicity or cytotoxic T cells must be checked, okay, but it is not the case in case of vitiligo, because in vitiligo what we found is that the regulatory T cells, which are characterized by the surface markers known as CD4 molecules, CD25 molecules, and intracellular marker known as FOXP3 for regulatory T cells. Are almost half as that of control. So that is the result that cytotoxic T cells, they are quite high in GB patient as compared to control. When we have seen this data according to the disease progression, we found that in active vitiligo patient also the regulatory T cells numbers are less as compared to the stable vitiligo. Okay, and CD8 T cells, cytotoxic T cells are quite high in active vitiligo patients. So one thing is clear that your Tregs are numbers are decreased, but there is functional defects also. Whatever Tregs are present in the vitiligo cells or in vitiligo, these Treg cells, they have functional defects in terms of releasing the immunosuppressive cytokines. For an example, interleukin 10, TGF beta. Okay, you have a FOXP3 which is also decreased in these patients. Cytotoxic T lymphocyte antigen 4 also it is being decreased. So what is a CTLA4? It is cytotoxic lymphocyte antigen 4 present on the T cell. Its main role is to do the T cell down regulation. So whenever there is a hyperactivation of T cells, CTLA4 molecules are synthesized and it will bind to the B7 molecules. That's how they engage the B7 molecules so that B7 molecules cannot react against your CD28 molecules. Okay, and it cannot then activate the T cells. But what we have found that the CTLA4 expression in vitiligo patients, it is very less. So there is lots of binding of B7 molecules with the CD28 and there is a hyperimmune response towards the T cell activation. So we have proven this that yes, your FOXP3 level is less and another protein which is associated with that nuclear factor of activated T cells. So this nuclear factor of activated T cells, they are also decreased and actually FOXP3 makes a complex with NF80s and then this complex leads to the expression of the cytokine gene. So what we have found out is that the cytokine genes are also less if you have less FOXP3 NF80 complex formation, right? So we did this experiment that the regulatory T cells we have isolated from the patient along with the cytotoxic T cells and helper T cells. We have done the co-culture assay and we found that there is high proliferation of your we can say autoreactive T cells because your regulatory T cells are having defects in releasing the suppressive cytokine. We found that your interferon gamma, which is the inflammatory cytokine, it has been released more by the cytotoxic T cells, which can readily do the apoptosis in the melanocytes and can result into vitiligo. So this we have already published in experimental dermatology that NF80C1 and FOXP3 plays a very important role in the functional defects of regulatory T cells in vitiligo. Now there is one study, animal model study, which suggests that if you use a vitiligo mouse model, and in this model if you transfer the functional regulatory T cells, right, the depigmentation rate in the mice, right, it decreases. Okay, so they have utilized this specific mice model known as H3TA2. So this mice model spontaneously generated autoreactive T cells, right, which identified tyrosinase as a autoantigen. Okay, so after 6 weeks of Treg transfer in the amount of 2 into 10 to the power 5 cells per ml, they could see good results. Okay, but the thing is that transferring or adaptive transfer of Tregs may be costly or may be requiring skills, right? So are there any mechanism? Sorry, just a minute. So are there any mechanism by which we can make this defective Tregs functional in the patient itself, right? Or maybe the differentiation of Tregs can be induced by some compounds. So a few years back, I proposed this idea of using probiotics and prebiotics. So we all know that probiotics are the good bacteria. They also play a very important role in modulating our immune system. Okay, probiotic will inducing the Treg. Okay, so whenever you have autoimmunity, let's say you have more of cytotoxic T cells and less of regulatory T cells, so probiotics and prebiotics can maintain the immune homeostasis. Means you can induce regulatory T cells so that you can nullify the effects of cytotoxic T cells. So let me explain you this particular slide. This will give some idea to the students also that if the problem exists in the immunological pathways, then how can be used by using the natural mechanism. So we all know that probiotics feed on a particular substrate known as prebiotics. These are nothing but the dietary fibers. Here there are examples like inulin, fructooligosaccharides, resistant starch and all. So once this probiotic feed on this prebiotics, they will release certain short chain fatty acids. So this short chain fatty acid examples are acetate, propionate and butyrate. All these three, what they do is that they are binding to the regular T cell. So as they will bind, it will allow the Treg cell expansion. Now this Tregs will increase in their numbers, okay? Lots of TGF. They will suppress the cytotoxic T cells, okay? So there are other mechanisms as well for this SCFs, like for an example, butyrate, they will cause SC inhibition in the epidermis. So this is SGSC means histone deacetylation, okay? So histone deacetylase is the enzyme which will so it Treg electroglobulin inducer of your interleukin 10. So component known as so this can help messages from melanocyte progression. So that in vitiligo patient, it can control to the melanocyte destruction. Okay. So I want to thank Sarb DSC to give me this project, and I also thank all vitiligo control patients. I thank our university, Ukatasadia University, for providing the infrastructure, and I especially thank Nanda Arts and Science College for giving me this opportunity to share some of my thoughts related to antigen processing and presentation with respect to. So I hello.
Thank you, sir. Thank you for sharing your knowledge with us.
Hello,
It
am I
would
audible?
be yes, sir. Thank you, sir. Thank you for your sharing your knowledge with us. It would be useful to us and which lead us to construct the best future. Thank you, sir.
Yeah.
Dear students, if anyone had doubt, please clarify your doubts with our chief guests.
Okay.
Dear students, share your feedback about the session.
Good morning, sir. Thank you for giving this wonderful presentation, sir. We have learned more information about this antigen processing and presentation, sir, by this webinar. You have discussed
Yes,
the articles regarding this, sir, that will give more information about this antigen presenting and processing, sir. Thank you for such a webinar, sir. Useful webinar, sir.
you're welcome.
Thank you, Ma. Now I welcome Ms. Jayasree from 3rd BSc to give vote of thanks.
Thank you, ma'am. Good morning to one and all present here. I would like to express my gratitude to all extreme delegates of the webinar for this presence and contribution to make this webinar a great success. I would like to thank our chief guest who honored this webinar with his inspirational thoughts, and I would like to thank our beloved principal, HODs, staff members, and my dear students for making this webinar as a grand success. Once again, I thank one and all present here. Thank you.
Thank you, Ma. Thank you, sir. Thank you to all.